Welcome back to Fun Facts Online. Today, we are going to dismantle the story you have been told about depression for the last 50 years and replace it with something far more complex and, frankly, far more hopeful. We are talking about the synaptic revolution, the NMDA receptor, and the neuroscience behind rapid-acting antidepressants like ketamine. For half a century, psychiatry relied on the monoamine hypothesis. This was the idea that depression was essentially a deficiency in feel-good chemicals, serotonin, norepinephrine, and dopamine. It was a neat, elegant narrative. If you were sad, your brain's fuel tank was low. The solution? Selective serotonin reuptake inhibitors, or SSRIs, like Prozac. These drugs were designed to top up the tank. And for many people, that worked. But for about 1/3 of patients diagnosed with major depressive disorder, and specifically those with treatment-resistant depression, that story was a lie. For these people, depression wasn't a mood slump. It was a neurobiological rigidity. Their brains were locked in a cycle of despair that serotonin simply couldn't touch. To understand why, and to understand the revolution happening right now, we have to look at the brain's hardware. Traditional antidepressants work on the brain's volume knobs. Serotonin doesn't carry the main signal. It just modulates the tone. Turning up the serotonin is like turning up the brightness on a television screen. It makes the picture more vivid, but if the picture itself is broken, if the wiring is faulty, turning up the brightness doesn't fix the image. This is why SSRIs take weeks or months to work. They are initiating a very slow, downstream cascade of effects. The real revolution began when we stopped looking at the volume knobs and started looking at the power cord. That power cord is glutamate. Glutamate is the primary excitatory neurotransmitter in the mammalian brain. It is responsible for over 90% of the synaptic traffic. Every thought, every movement, every sensation you are experiencing right now relies on glutamate firing across a synapse. In a depressed brain, particularly one subjected to chronic stress or trauma, the glutamate system is out of balance. Chronic stress floods the brain with cortisol. Now, cortisol is useful in short bursts, but over time, it acts like a toxin to the delicate branches of your neurons, specifically in the prefrontal cortex and the hippocampus, the areas responsible for thinking, mood regulation, and memory. Under this chemical assault, neurons physically shrivel. They lose their dendritic spines, which are the tiny receivers that catch chemical signals. The brain's hardware literally disconnects. You aren't just sad, you are operating with degraded circuitry. This is where the star of our show enters, the NMDA receptor. The N-methyl-D aspartate receptor is one of the most sophisticated machines in biology. It sits on the surface of your neurons, acting as a gatekeeper. It is an ion channel, a pore in the cell membrane that lets charged particles flow in to create an electrical signal. But the NMDA receptor is special. It is a coincidence detector. Under normal resting conditions, the NMDA receptor is blocked by a magnesium ion. Picture a bottle with a cork in it. The magnesium is the cork. Even if glutamate floats by and tries to activate the receptor, the channel stays closed because that magnesium cork is stuck tight. For the channel to open, two things must happen at the exact same time. First, you need the chemical signal. Glutamate must bind to the receptor. Second, you need a strong electrical signal from the neuron itself. Only when both happen, strong activity plus the chemical signal, does the electrical charge pop that magnesium cork out. When that happens, calcium rushes into the cell. This calcium surge is the biological signal for neuroplasticity. It screams to the cell, "This connection is important. Strengthen it." In depression, this system is glitching. There is often an accumulation of ambient glutamate outside the cells, creating a constant background noise. This leads to tonic activation, exhausting the neurons, causing them to shrink and stop communicating. The brain loses its plasticity. It gets stuck in a rut. Enter ketamine. Ketamine is an NMDA receptor antagonist. That means it blocks the receptor. Now this sounds completely counterintuitive. If depression involves a loss of synaptic connections, and the NMDA receptor is responsible for making connections, why on earth would blocking it help? Why would a drug known as a horse tranquilizer or a club drug wake up a depressed brain? The answer lies in the disinhibition hypothesis. The brain is a delicate balance of gas pedals, excitatory neurons, and brake pedals, inhibitory GABA interneurons. When ketamine enters the brain, it doesn't just block everything randomly. It preferentially binds to the NMDA receptors on the GABA interneurons first. It targets the brake pedals by blocking the NMDA receptors on these inhibitory cells. Ketamine stops them from firing. It cuts the brake lines. Suddenly, the excitatory neurons, the gas pedals, are disinhibited. They fire rapidly. This causes a massive, transient surge of glutamate release in the prefrontal cortex. Here is the magic trick. This sudden flood of glutamate looks for receptors to bind to. But remember, the NMDA receptors are blocked by the ketamine. So the glutamate has to go somewhere else. It binds to the AMPA receptors instead. AMPA receptors are the workhorses of fast signaling. The activation of these receptors causes a strong electrical surge in the neuron, opening distinct voltage-gated calcium channels. Calcium rushes in through a back door, bypassing the blocked NMDA receptor entirely. This calcium influx triggers the release of BDNF, brain-derived neurotrophic factor. Think of BDNF as Miracle-Gro for the brain. BDNF activates a protein kinase called mTOR, which is a master switch for protein synthesis. When mTOR is flipped, the cell immediately starts building new synaptic proteins. Within hours, those shriveled neurons begin to regrow their dendritic spines. The neural circuits in the prefrontal cortex physically reconnect. This is what scientists call synaptogenesis. It is the biological basis of the rapid-acting antidepressants. We are physically repairing the hardware so the software of your mood can run smoothly again. Now, science is rarely settled, and there is a fascinating subplot here known as the metabolite mystery. Researchers at the University of Maryland discovered that when ketamine breaks down in the body, it turns into a metabolite called hydroxynorketamine, or HNK. In mouse models, HNK appeared to produce antidepressant effects without blocking the NMDA receptor and, crucially, without causing the trip or dissociation associated with ketamine. This suggests that perhaps the famous NMDA blockade is just a side effect, and the real cure lies in this metabolite stimulating AMPA receptors directly. This has sparked a gold rush to develop drugs that mimic HNK, antidepressants, that work in hours but don't make you hallucinate. However, for now, in humans, the NMDA receptor remains the primary proven target. Speaking of the trip, let's talk about the controversy of dissociation. When patients take a sufficient dose of ketamine, they experience a separation of mind and body. Time dissolves. The ego, the sense of I, fragments. For pharmaceutical companies, this is a bug. It's a side effect called a psychotomimetic effect. It makes the drug hard to administer because patients have to be monitored in a clinic for 2 hours to ensure they don't hurt themselves while dissociated. This is why we have drugs like Spravato, or esketamine, which is a nasal spray requiring a strictly controlled risk evaluation protocol, or Auvelity, a new pill that combines dextromethorphan with bupropion to block NMDA receptors more mildly without the intense trip. But for many clinicians and therapists, the dissociation is not a bug. It's a feature. They argue that the trip provides a time out from the relentless, rumination-heavy narrative of the depressed brain. It offers a psychological reset, allowing patients to view their trauma from a detached, objective perspective. Patients often report a lifting of the heavy blanket of depression, a subjective experience that correlates with the biological repair of the synapse. It's a debate between the bioengineers who want a clean pill and the psychotherapists who value the experiential breakthrough. The truth is likely in the middle. The biological repair works regardless, but the psychological experience may help sustain the recovery. There was another plot twist in 2018 that scared everyone. A study out of Stanford showed that if you gave patients naltrexone, an opioid blocker, before giving them ketamine, the antidepressant effect disappeared. This terrified the medical community. Was ketamine just a fancy opiate? Were we just getting people high? Subsequent research clarified that while ketamine does interact with the brain's endogenous opioid system, it isn't an opioid in the traditional sense. It doesn't stop your breathing. It seems the opioid system is a necessary cofactor, a helper, for the NMDA mechanism to engage the plasticity machinery. So where does this leave us? We are entering the era of psychoplastogens. This is a new term for drugs that produce rapid and sustained neuroplasticity. The pipeline is crowded. We have drugs like NRX-101 attempting to treat bipolar depression without the trip. We have nitrous oxide, laughing gas, being investigated for similar effects. We are looking at selective antagonists that target specific subunits of the NMDA receptor to separate the cure from the hallucination. The discovery of the NMDA receptor's role in depression is arguably the most significant breakthrough in psychiatry since the 1950s. We have moved from a model of chemical imbalance to a model of circuit failure. We are no longer just topping up the fluid in the tank. We are rewiring the engine. By targeting the NMDA receptor, we are effectively jump-starting the brain's natural ability to heal itself. Whether through the dissociation of an infusion or the silent plasticity of future pills, the glutamate revolution has finally given the depressed brain a way to reconnect with the world. That is the new reality of rapid-acting antidepressants. Thanks for listening.